A patient's fully healed surgical scars spontaneously pull apart. Deep beneath the skin, internal capillaries violently rupture, spilling blood into the surrounding tissue. Biologically, humans are trillions of delicate cellular bags composed mostly of water. To withstand gravity and the mechanical forces of a beating heart, those bags must be bolted together by a specific internal architecture. That architecture is defined by L-Proline. Accounting for up to 12% of the body's total amino acid inventory, it serves as the master building block of human structural resilience. We often treat our physical solidity as a guaranteed passive state. In reality, it is an active, continuous architectural defense, engineered entirely by the rigid geometry of this one chemical. 19 of the 20 standard amino acids used to build human proteins behave like flexible pieces of string, bending whichever way the body needs. L-Proline operates differently. It is the human genome's only native alpha amino acid. Instead of a flexible tail, proline's 3-carbon side chain loops backward, fusing directly with its nitrogen atom to form a rigid pyrrolidine ring. Biochemists call this a steric constraint. It physically forces the molecule into a permanent, unyielding angle of -65 degrees. Folding standard flexible proteins requires the body to burn significant amounts of energy to force those links into position. Because proline is already locked into this angle, it lowers the thermodynamic resistance to folding, providing the structural foundation needed to construct dense, load-bearing mammalian scaffolding. A healthy adult carries roughly 75 grams of this scaffolding material. 85% of it is permanently bolted down in what pathologists call the skeletal vault, locked deep inside mature bones, tendons, and large vascular walls. The remainder circulates as a highly mobile reserve pool within the blood plasma. When a patient suffers a major burn or undergoes extensive surgery, the body initiates a localized response known as a wound siphon allocation wave. Driven by inflammatory signals, local fibroblasts at the injury site sprout millions of specific SIT1 transporters on their cell surfaces. These function like microscopic vacuums, aggressively pulling free-floating proline out of the passing bloodstream. The cells take this commandeered proline and rapidly braid it together to build a tight, triple helical molecular rope known as collagen. This forms the immediate structural foundation for emergency scar tissue. Clinical models demonstrate that if circulating plasma proline levels drop below 80 micromoles per liter, this rapid response factory runs out of raw bricks. The structural scaffolding stalls, leading directly to catastrophic surgical wound failure. This entire collagen factory has one fatal vulnerability. To lock that triple helical rope together permanently, an enzyme called prolyl hydroxylase must add oxygen atoms to the proline. This enzymatic step is strictly dependent on vitamin C. It functions as the necessary cofactor to maintain the iron atoms at the enzyme's core, and without it, the entire assembly line stops. Without that vitamin C bridge, newly manufactured collagen lacks the extensive network of stabilizing hydrogen bonds, causing it to physically melt at 37 degrees Celsius, normal human body temperature. This widespread matrix collapse is the physiological reality of scurvy. This delicate processing machinery must constantly adapt to different biological demands. During pregnancy, a developing fetus will aggressively mine the maternal plasma pool, pulling proline across the placenta against a steep concentration gradient to secure the raw material required for fetal bone development. Conversely, as the body ages, the enzymes that process and cross-link this molecule naturally decline. The visible signs of aging, from sagging dermal layers to the stiffening of arterial walls, are the direct biological decay of this exact structural processing network. If a lack of proline causes the matrix to melt, an overabundance creates an entirely different crisis. In an inherited genetic condition called hyperprolinemia type 2, the metabolic recycling plant completely breaks down, causing proline to rapidly accumulate. Trapped with nowhere to go, these excess structural molecules build up to extreme toxic levels inside the central nervous system. In infants, this buildup turns a foundational building block into a neurochemical poison, triggering severe seizures and developmental malaise. While infants suffer from this inherited genetic accumulation, healthy adults can accidentally trigger a similar transport failure through acute isolation overdose, typically by ingesting massive, unregulated scoops of pure L-Proline supplement powders. This sudden influx completely overwhelms the CYT1 transporters lining the intestines. The unabsorbed powder sits in the gut and creates a severe osmotic imbalance, aggressively pulling water from surrounding tissues to dilute the chemical spike, resulting in extreme gastrointestinal distress. The human architecture demands precise balance. Dumping an excess of molecular bricks onto a biological construction site does not force the body to build stronger tissue. It simply crushes the metabolic transport workers attempting to process the load. To verify a patient's structural integrity and avoid these crises, clinical laboratories rely on liquid chromatography tandem mass spectrometry. This technology physically separates the complex mixture of molecules in the blood and weighs the fragments to confirm their exact chemical identity. This line graph demonstrates a diagnostic trap known as the non-fasting surge. If a patient eats a high-protein meal within 4 to 6 hours of their blood draw, their circulating plasma proline artificially spikes by up to 40%. If a physician looks at this inflated data without knowing the patient's dietary timeline, they miss the reality of the situation. That artificial postprandial spike completely hides a dangerously low structural baseline, leading a doctor to believe a starving patient is perfectly healthy. A second blind spot is the hemolysis illusion. If a phlebotomist uses a needle that is slightly too small or shakes the collection tube violently, the physical shear stress rips the red blood cells open, spilling their highly concentrated internal proline directly into the testing sample. Interpreting advanced mass spectrometry results is clinically useless in a vacuum. Accurate diagnosis requires strict diagnostic stewardship and a complete understanding of the physical reality of how that specific blood sample was collected and handled. Deep within cellular metabolism, there's a final paradox at play, known to biochemists as the proline apoptotic shunt. When a cell faces severe, unremitting stress, such as extreme suffocation or starvation, it becomes desperate for energy. To survive the immediate threat, the cell makes the drastic decision to cannibalize its own internal proline stores for a quick hit of ATP. As electrons are transferred directly into the mitochondrial respiratory chain, the process generates a sudden surge of reactive oxygen species. These toxic byproducts eventually damage the mitochondria itself, triggering the release of cytochrome C and activating the cell's internal suicide program. From anchoring the triple helical collagen rope, to providing the hydroxyproline substrate, to acting as an osmotic protector, to forcing structural kinks, to fueling mitochondrial oxidation, proline dictates human structural integrity. Next, we will examine the polar pioneer of one-carbon metabolism, serine. Our physical resilience is a highly delicate, dynamic state. The cellular bags of water that make up our architecture are held together by a single rigid molecule, constantly dancing on a razor's edge between keeping us intact and burning our foundation to the ground.